Hi everyone, and welcome back to the Computomics podcast. Last week we mentioned that we will be discussing the Pantograph project, which is our effort to make a contribution to the fight against COVID. And today we are interviewing Josiah Seaman, who's an integral member of that project and a great collaborator. And he will walk us through the focus of the project, the reasoning behind it, and how you can help. And if you are a computational biologist or a bioinformatician, or if you are a software developer and you want to put your skills to good use, this is your chance. He'll mention it again, but the link is graphgenome.org. And please join us. Thank you for listening, and thank you to Josiah for being great to interview and such a pleasure to talk to. Now, here's the episode. Hi, Josiah, and thanks for joining me today. It's going to be such a pleasure to introduce everyone to our Pantograph project. I know that you're an amazing collaborator of Computomics, and you are spearheading this campaign, and you are very passionate about this campaign and about ways that it can empower people to contribute to the fight against COVID. And I just want to go ahead and jump right in. Maybe you can tell everyone what is the basis for the Pantograph project, what is its mission, and where are you going with it?
Yeah, so I'm kind of a big picture person. So I think what I'll do is start at the biggest. First of all, I would say genetic engineering and knowledge of genetics has immense potential. And though we've invested money into starting to develop the science for the most part has not touched everyday citizens at all. GMO is banned in most of Europe. We were working at Kew Gardens on ash dieback resistance to save the ash trees, and people were very hesitant to even change one gene to stop the extinction of the most common hedgerow tree. So there's still a lot of resistance there, and I understand where that's coming from. As I said, there's technologies that have downsides, and so then if we zoom in a bit more to what makes genetics challenging, it's really about the idea of transferring knowledge from one study or one instance to another. And that's all done through taxonomy and the study of comparative genomics. And then if we zoom in more on comparative genomics, we have what's called a reference bias problem, where, for example, the human genome was constructed using largely European people, and it does not do well in genetic analyses on African people, because the Africans' genomes are aligned to a European genome, and the things that don't match up often get discarded in the analysis. So this reference bias problem is what we're trying to solve with graph genomes. And we think it could be a really powerful tool so that you can go out and take many individuals of a species, sequence them, and get a bias-free global picture of the genetic diversity in that species. That's relevant for plant breeding, animal breeding, disease resistance, viruses, all of it. So then that was really the motivating goal for the Pantograph project. And then once COVID-19 really came out, and once I started taking it seriously, I realized, we're studying sequence diversity within a species, and the sequence diversity of a virus is very important for two big reasons. One is that the morbidity, the mortality rate can change based on mutations. Oftentimes it actually goes down. So for example, Ebola has become has a lower mortality rate than it used to. But that can also make it more infectious. So it's a bit paradoxical.
Right.
And the second thing has to do with resistance. So the genetic diversity of, for example, the common cold, if I can kind of simplify things a bit, the genetic diversity is so high that it's not really possible to create a vaccine for the common cold, because it's actually many different things, and even within those viruses, there's a great degree of genetic diversity. What I got concerned about with SARS-CoV-2 is that the ability for a virus to mutate and evade something like a vaccine is proportional to its population size. So if you think about it like a deck of cards, SARS-2 gets a new deck of cards every time it infects a new person. And so the odds of getting that sort of 1 in a million royal flush get proportionately higher as more people get infected. So we know, for example, seasonal flu, influenza, is an RNA virus, and it has a high enough mutation rate that we need to update the vaccine about once a year. Well, if that virus, or for example, SARS-2, infects 10 times more people, you're looking at a 10 times larger potential to evade the vaccine. And so that's really kind of what we're focusing on, is this kind of mid-game where we've contained with quarantine, we've developed a vaccine, and the vaccine's being disseminated through the population. We're going to monitor the genetic diversity across the whole planet and ask the question, is there any virus variants, little subpopulations, where they could be resistant to the vaccine while the rest of the world is not? Because if that happened, then, and we didn't notice it, then what would basically happen is you'd have a selection sweep that would wipe out all of the non-resistant variants, and so only the resistant variants would be left, which could then spread through the population again. And this actually applies to acquired immunity too. So it's an unlikely scenario, but when you're dealing with something on a global scale, it makes sense in terms of the investment of resources to invest resources in covering that 1 in 10,000 chance that would be really costly if it ever cropped up. So we're not the most important thing going on right now, but we're covering one of these edge cases, and I'm trusting other professionals to cover, to cover the areas that I don't understand, that I don't have the skills for. I don't know how to make masks or ventilators or any of these things. I know software, I know graph genomes, so I'm trying to put my skills to work to save as many people as possible.
So when we talk about features of graph genomes then, since it seems to be your wheelhouse, what are the aspects of graph genomes you think are for these scenarios for them to be useful and provide the kinds of answers that will lead to rapid action, but appropriate action?
Yeah. So graph genomes cover an area of sequence variation that other tools don't, which has to do with rearrangements. So it looks like, for example, the 1,000 Genomes Project on humans, there's about as many nucleotides that are affected by what we call SNP mutations, single nucleotides, so single letter changes, as are affected by rearrangements or large-scale structural variation. So one that I think people would recognize, which is easy to spot, is Down syndrome, is a duplication of an entire chromosome, which is a kind of an extreme example, but it's an example. I think variation, we're all running on the same hardware, so it applies to everything from viruses to fungus to trees to people. So pretty much anywhere where you would use genetic engineering, I prefer people use graph genomes over the older methods. because so I sort of look at it as we're developing the next generation of tools, the next generation of techniques that cover these edge cases that have not been covered before.
And what kind of people are you trying to get involved? Who do you feel like who's working with you now? Because you guys have some really great collaborators. I know in Computomics we're working with you, but there's also kind of a world global consortium, and they're really amazing people. So what kind of people are all working together on this current project?
Yeah, I mean it's really amazing. We've got Asia, Africa, Europe, North America. I'm not sure if we have anyone from South America at the moment, but that could change. I think it's something like 20 different countries within my own team, and I have lost track of how many countries in the total biohackathon, because this is a whole world event. This is, in a large sense, the third, full world event that we've ever had. So it's exciting to be surrounded by so many talented people. I'm mainly pulling in computer scientists. If people have wet lab experience, they should probably be, testing people, developing reagents, things like that. For us, computer guys and gals, we a lot of them don't necessarily have the biological background to understand the full picture, but with open source software collaboration, we can define an issue, basically a single ticket that says, if you can make the program go from this state to this state, that will be a tangible improvement. And all that requires is software programming knowledge. It doesn't really require deep biological knowledge. And so I'm trying to give an opportunity for people who otherwise would feel kind of powerless, just sitting at their homes wondering if their loved ones are going to get sick. I'd prefer to be doing something, even if it's a small thing. Having something to do that could benefit, I think, is an improvement.
Definitely. And would you say that there's still time for people to join? I know it's going on this week
Yeah,
from until the 11th of April. So if we put this out, is it still possible for people to join you?
absolutely. I'm actually planning on dedicating several months to this, just because I feel like that's a proportionate response. I've kind of rearranged part of my life to to really try and contribute positively to this. So I'm not planning on stopping. We'll have an end of hackathon celebration, but then I'll keep going after that, and I think that some of our team members are going to keep going. It's really just a question of what people can, what people can volunteer. We're actively pursuing funding right now to be able to pay our volunteers and to maybe grow this into a whole lab. I think if you look at historically, where we were spending our research money in 2019, it's a tiny, tiny fraction of the world's economy was devoted to science. And now we're looking at, $6 trillion impact for just the US alone, probably looking at maybe $20 trillion worldwide disruption. With $20 trillion, we could have, had cold fusion by now and immortality. So, uh, so it's like, what is a bad management of resources? It's saving $1 billion on NIH funding in order
Yeah.
to have a 1% risk of a $1 trillion loss. Like, that's just bad business, right? It's bad logic. But I mean, I've been banging this drum for 10 years, and and people really need an example in their lifetime to make it real. So, I mean, I could easily see science funding being 10 times greater than, globally, than what it was last year, maybe in the next 3 to 4 years, if there's a positive impact to this. And so I'd say, well, let's keep going. I mean, we were doing a good thing before, and now it's just all that more important. So yeah,
Yeah,
get volunteered, and we'll see if we can scale this up. I can't make
yeah,
any
that makes me
promises.
so happy.
The future is really uncertain right now, but I wanna be there when the future happens.
When the pendulum swings in the favor of science, that would be a great, that would be a great metaphor or outcome of this decade. If 2020 was the beginning of the pendulum going in the favor of science, then I think that would give me a little measure of peace to go to sleep at night.
Yeah, yeah, and there's upsides and downsides to everything, but the way I see it is we've given business a lot of resources and a lot of, I almost want to call it like democratic freedom, almost like a Bill of Rights sort of stuff, in order to be able to fully actualize what business is capable of. And so we've kind of reaped the benefits of big business, and there's been this ongoing talk about uh, what is it called, efficiency of scale and those sorts of things. Well, part of efficiency of scale is that a virus which replicates exponentially can hit the entire world, right? That kind of
Yeah.
comes as a package deal. And so, if the pendulum swings in favor of science, for example, we might be a little bit more inefficient. We might be more navel gazing. I don't know, but I'm sure there are some downsides. But I know that the upsides can be absolutely immense. We can end world hunger. We can, get all clean energy. There's a lot of good things that I think that science, research, and engineering have to offer that have been blocked so far.
Yeah. Well, to kind of get you support to get the ball rolling, just tell us quickly how people can get involved. And you mentioned already what you're looking
Mm-hmm.
for, and sort of, what is the call to action for anyone
Yeah,
interested?
so we have a website, graphgenome.org. So graphgenome.org. And that's got the Pantograph website up there right now. That kind of details what we're trying to accomplish with our browser. And that also has a Get Involved section with a link to our Slack. We're using Slack as our central point of communication. And so you just go into Slack, introduce yourself, say, these are my skills. I want to get involved. It could be, funding and grant writing. It could be management. It could be software. It could be a deep understanding of the biology. And then we'll get you set up with one of our teams, get you on an issue that you can, add your brick to the construction project, and then basically all of those communally get brought together into a project that's a lot more grand than what any person could accomplish by themselves.
Yeah, fantastic. Thanks so much. I really hope that people who maybe thought that their expertise wasn't necessarily relevant, feel like they now have an avenue to make that experience incredibly
Mm.
relevant, useful, long-term, actionable. All those things I think are socially
Mm.
empowering, intellectually empowering, of course. So I am on a personal level so impressed by you. I can see, yeah, why York suggested that we talk. You're amazing. What a wealth of information. Thank you so much for your time.
Well, we're
And
not on.
I
You
really
can't
hope.
see me blushing. I, I, I don't want to. Yeah, it's mostly been about just having a vision and putting a team together. We've had a lot of really great contributions. That's a lot more than what I could do on my own. Um, and yeah, I mean, I thought 2 months ago my skills were totally irrelevant to the situation. And so some of this has just been a change in mindset for me. And so I know whatever skills people have, there's a way that they can contribute.
That's so great. That really means a lot. So yeah, I really look forward to you guys recruiting more and doing more and moving forward with your vision. You seem to be incredibly on purpose. That's one of my favorite things is people who are doing what just seems what they're meant to be
Mhm.
doing. And I think the more we have of that in the world, the better the world will be. The more people are on purpose, I think people can live with incredible satisfaction, gratitude, mission, vision, and yeah. So thank you so much, Josiah, and I really hope we get to talk
Me
again.
too.
And maybe I'll check in with you again. Maybe you can give me updates, whatever you have time for.
Yeah, I'd love to. This has been really great talking with you. Thank you for letting me share my
Yeah,
perspective. Uh,
yeah. Thanks a
all
lot.
right.
Okay,
Have a
bye.
good evening.
So thanks everyone for listening to our second episode in our interview with Josiah. If you're interested in contributing, please go to graphgenome.org. You can also visit our website, computamics.com. Please subscribe to our podcast if you'd like to hear more and visit our website for blogs and updated information. Thanks so much.